Thank you so much for taking the time this evening to listen to our very honored guest, Professor Patrick Maxwell from Cambridge. I'll give you a little short introduction to him in a little while. But for now, I think it would be appropriate for me to invite Professor Ron Zeman up to the stage here. Professor Zeman is a personal friend of Professor Patrick Maxwell.
Thank you, Terry. Terry is going to introduce you, Patrick, formally, but I just wanted to have a few seconds to say a few words about how pleased I am that you were able to make the time to come to Hong Kong. I am very conscious of the fact that these Hong Kong-Cambridge links are important. I know that in the very early days, Professor Lord Butterfield was here for a considerable time, spent a considerable time here working with Professor Sir David Todd. And then he was followed by Professor Sir Keith Peters and then Professor Sir Patrick Sissons, unfortunately, who passed away last year, Patrick. And we now have another Patrick as our Regius professor. So this is continuing in a most amazing link between our two universities, which I, as someone born in Hong Kong and having spent most of my professional life in Cambridge, treasure very much. So over to you.
Thank you. Professor Ron Zeman is also the representative of the Hatton Trust, which is one of the two sponsors of this series of lectures. Perhaps you will allow me now to give you a short bio of Professor Maxwell. I've been told to keep this really very short. Professor Patrick Maxwell is the Regius Professor of Physic and head of the School of Clinical Medicine at the University of Cambridge. The professorship was founded by King Henry VIII in 1540. He graduated from Corpus Christi College, Oxford, in 1983 with a first-class honours in physiological sciences, and there did his clinical training at St. Thomas's Hospital, where he won the Mead Medal in Medicine and the Cheselden Medal in Surgery. He undertook postgraduate clinical and research training in nephrology and general medicine at Guy's Hospital and in Oxford, and was appointed as university lecturer and then reader at the University of Oxford. In 2002, he moved to the professorship of nephrology at Imperial College, followed by the chair of medicine at University College London in 2008. In 2012, he was appointed to his present offices of Regius Professor of Physic and head of the School of Clinical Medicine of the University of Cambridge. The Regius Professor of Physic is head of the School of Clinical Medicine at the University of Cambridge and director of Cambridge University Health Partners, which is the Academic Health Sciences Center the Academic Health Sciences Center for Cambridge. We are very privileged this evening to have Professor Maxwell here with us as our distinguished visitor to deliver the annual WYNG-HATTON Lecture. He will be tackling the following theme, as you see up there: how medicine is changing, a perspective from clinical sciences. So, without further ado, will you please welcome with me Professor Patrick Maxwell to the podium.
Well, Terry, thank you very much for that nice introduction. So this is the first time, I think, that I've lectured to a law faculty, and it's a terrific privilege to be here in Hong Kong with you. So I think something is going to change in medicine, but I would have to say it's changing rather slowly. So if you're going to go to sleep for the rest of the lecture, the answer to the question, how is technology changing medicine, the answer is very slowly. And to illustrate that, these are pictures from England about 40 years ago, I think, and they would actually look rather similar today. So at the bottom is the local branch of the National Westminster Bank in the small town where I grew up. And I remember, as I'm sure all of you do, how you used to run your bank account. Now, if you wanted money, you would have to queue up. If you wanted to pay a check, you'd have to queue up. You'd write a check to people. And I guess most professional people would go into the bank once a week. And if you wanted to arrange a loan, then you had to have an interview with a bank manager. And nowadays, I mean, I have not been into a branch of a bank, I think, for 5 or 6 years. And I do everything over my mobile phone. Now, next door to the bank, there were shops. And, you know, most people would go to the shops most days. And of course, all that has changed quite dramatically. Most things we do with our phones. And I have never liked shopping, and all the shopping I do now, I do on Amazon, and the parcels arrive within 24 hours. I changed banks. I was right at the beginning, or I didn't like going into the branch of the bank, and actually I opened an account with Hong Kong and Shanghai Banking Corporation, which had this branch First Direct, and you could do all your banking over the telephone. So in banking or arranging travel or doing your shopping, that has changed in the most fundamental way over the last 30 years. And medicine, of course, is a bit hard because I'm a practicing doctor, so, you know, classically we don't really see change creeping up on us. But the way that my patients interact with me in the hospital has not changed very much at all. We still write them letters, which we send them in the post. We still ask them much the same questions. In many hospitals, and many of you who are doctors probably still write paper notes. So I'm going to talk to you briefly about 3 things this evening. First of all, electronic health records. Second, a brief vignette about imaging. And thirdly, I'm going to talk mostly about genomics. So if you, I don't know how it is in Hong Kong, but most hospitals in the UK still have still have medical records departments that look something like this picture. And there are some very serious problems about paper records. So first of all, in maybe as many as 1 in 20 visits to the hospital, the patient records don't catch up with the patient. So you see the patient without the record, and you have to start from scratch. Actually, sometimes that makes it easier, because usually the doctor's handwriting in the notes is rather illegible. So it isn't necessarily even an advantage having the paper notes there. Now, like many hospitals, we've embarked in Cambridge on an electronic health record, and we introduced that now about 3 years ago. We did it with this provider called Epic, and we were the first hospital in Europe to implement this American electronic health record. And I can't pretend it was easy. So overnight, basically, the hospital switched from being an almost entirely paper hospital to an almost entirely paperless hospital. And it's been very interesting to watch that. So for the youngest doctors in the hospital, this was not a problem at all. They found it quite straightforward. For the slightly older doctors like myself, the truth is that a few years later, we're still struggling with this system. It really was a fundamental change in the way we communicate with patients. And I think what is quite striking is that within a few weeks, if you stopped a doctor on the corridor of the hospital, I think the great majority of doctors would have said that although this system is far from perfect, it enables them to do things that they couldn't do before. So in my specialty, which is nephrology, looking at the blood results is really important. This system now means that in a secure way, using my tablet or phone, if I'm traveling, I can look at the key results on patients without having to speak to anyone by telephone. And in nephrology, that is a big step forwards. And it also means that all of the nursing notes, all of the notes from anyone in the hospital are in a single shared record, and that also is fantastic. And there's some really good things, like if we wanted to take a photograph of something about a patient in the past, you had to fill in a form and send them off to medical photography, where a professional photographer would take a photograph, having got their permission, and put it in the record. With this system, you can just have an app on your mobile phone, and you can take a picture, and that is not retained on your mobile phone. It goes straight into the patient record, so there's no problem about confidentiality. Ask the patient for consent, and then it can be there on their record. And just a few weeks ago, I went to a very interesting grand round in the hospital where a patient had been traveling, I think, in Africa. The dermatologist had seen them with this swelling, and they reported the case at the grand round because when they incised this in the operating theatre, a rather horrible larva of some insect crawled out of the wound. And they were able to photograph this and put it straight into the medical record. So that is a fantastic advantage. And this is undoubtedly better for patients and the organization. So you can see here that we've had a reduction in all sorts of errors. I think balancing that, one would have to say that we have also been able to create totally new kinds of errors because of the problem of doing things by computer. But I am convinced that this has removed all sorts of errors. So you can see that there's a decrease in errors on prescribing antibiotics that people are allergic to, much better prescription about sedation, and the rapid response team can detect from people's observations when they should go and see someone without the doctor having to be called. And you should always take things like this with a pinch of salt, but in most hospitals, many aspects of process do not work perfectly. And this overall approach enables us to work much better in terms of making sure that processes work quickly and smoothly, and it reduces the variation between individual doctors. And you can see that according to this study, there's been a substantial reduction in mortality, 42%, from patients coming into the hospital with sepsis. Now, it's also increased efficiency in the hospital. I'm on the board of the hospital, and we, like other hospitals in the UK, we are underfunded, and we have a large financial deficit. And you can see that this slide, which was put together by the electronic health record team, tells you that we are saving quite a lot of money having put the electronic health record in place. But what I can tell you is, like many other things in medicine, actually this hasn't saved the hospital much money. We've probably spent more money because when processes improve, we don't actually get rid of staff in a UK hospital, but it does liberate the staff to do other things, like spend more time with the patients. So I think it's good for communication, but at least in a UK setting, I would say this has not saved us money. And working as a doctor in a paperless hospital, I would say that this has been a considerable advance. It has huge potential, but it still has a very long way to go. I wouldn't want anyone... Is there anyone from Epic in the audience? Okay, well, that's a good thing, because I would say that Epic... the front end of Epic is not like an app on your iPhone. This is not an intuitive system to use. Often, you have to say, to close an encounter with a patient, I have to go through 6 steps that are basically the same every time, and it's completely unclear why I have to do that. So An advantage of this system is that EPIC does allow a user like our hospital to modify it, but we have a tiny team working on improving this for our hospital. And it is an American design system, so a number of the things do not read across easily into a UK hospital. It's used by most of the big academic health science centers in the US. That's a huge advantage because it means that in terms of research, there's the possibility of sharing information across different systems and bringing a huge amount of information into play. But a colleague of mine at Harvard estimates that they have about 1,000 full-time equivalents working across the partners' health system, adapting EPIC to their circumstances and making it suitable for research. And so in Cambridge, I would say that this is improving our processes, but it is not yet there as a system that can deliver much research usefully. I think it underlines another important problem, which is that I think to do really good research on health and healthcare, you need to access the record of everything that happens to the patient, really from when they're born, ideally even when they're conceived, through to when they're dead. And that means you really need a record that reads across into primary care. Ideally, it would read across into education, social care, so that you had access to everything. And the UK should be a fantastic place to do that because we do have a comprehensive healthcare system that is open to everyone and is accessed by almost every system. But there is a problem, which is that the way that primary care works in the UK, people are basically independent practitioners and they all own their own record. And a fundamental challenge when we handle patient records, which might be interesting to a legal audience, is we really worry about confidentiality and consent. So in the UK, one of the interesting illustrations of this is if I want to find patients from the electronic health record to invite them to take part in a research study, I'm not allowed to do that unless they have already consented to their record being used for research. So you need consent for consent. And we're aiming, of course, in a hospital like ours, to get consent from every patient as they come through the door for everything, but you'll also understand that consent has to be tailored to what you're going to do with the information. So in thinking about this in a UK environment, my argument has been that we really need to solve the transfer of information for patient care. So certainly patients worry about what we will do with their record from a research perspective. They don't worry at all about us using it for patient care. And at the moment, if a patient comes to see me in the renal outpatients, I can't see what's happened in primary care with their GP, and I don't know what drugs they've got from their pharmacist. And that's a surprisingly difficult problem. So trying to get an accurate list of the medicines that a patient is actually taking is fundamental to looking after them well. And this may be different for an audience like yourselves, but trying to get a patient to tell you exactly what medicines they're taking, certainly when they started and finished them, is almost impossible. So we have a long way to go on that, and it is tied up with regulation, confidentiality, and I think it's very important to appreciate that this is a different task from my bank having information about my finances on the phone, or Amazon delivering what I want from the shops. Because an encounter with a hospital is really, really complicated. And there are lots of things there that doctors still use, all sorts of things to describe and explain that we can't. And I think it is an error to think that current machine learning can turn what doctors write into a system into useful information that can be easily turned into meaningful knowledge. But we are ambitious in the UK, and there is a big national project now called Health Data Research UK. Cambridge is a spoke in this. We are actually focusing on how to interface genomic information with clinical phenotype. And I'll come back to genomics in a minute. But a good illustration, I think, of what can be achieved is this commercial company, Flatiron, in the United States, which has done a project of collecting community and hospital information on patients with cancer and what they've been treated with, so that you can work out the connection between outcomes and treatments in the real world. And my understanding is that that has already delivered regulatory approval for some cancer medicines in cancers where they were not previously approved. I'm going to touch very briefly now on imaging, because this is an area where the tests that we do have enormous amounts of information that is not currently used completely for clinical care. And I think this is an area where machine learning and artificial intelligence could make quite a big difference quite quickly. I think it will also decrease the value of radiologists. So are any radiologists in the audience? So, you know, if I was a radiologist, I would be worried about my future at the moment. The story I'm going to tell you is about cardiac catheterization. Like many medical stories, the beginning of this was extraordinary. There was an eccentric guy called Forsman. He was actually a urologist, and he had this vision of being able to work out what was going on in the heart by inserting a tube into the heart. So he took a urology catheter, and his plan was to insert it into his own vein and then float it round into the right ventricle. And he persuaded a nurse to help him, and she said, "I'll only let you do it if you do it on me, not on you." And this was in pre-Nazi Germany. And he actually tricked her in the operating theatre, and he did it on himself after all. And then they walked up to the X-ray machine, and he was able to show that he had got the tube into his heart. And that was then followed up in the United States, where they developed the first cardiac catheterization lab. And the two of them won the Nobel Prize jointly for that. And since then, cardiac catheterization really revolutionized the treatment of ischemic heart disease. But it's a difficult and expensive investigation that needs a cardiologist or a radiologist to do it. And it costs a lot of money. And it probably leads to quite a bit of over-investigation and over-treatment. Now, quite a big breakthrough was recognizing that you can get quite a lot of information from doing a CT scan of the coronary arteries and just injecting contrast. And so then you need much less expertise. But there's a problem in that most of the narrowings in the coronary arteries actually have some calcium in, and that produces artifact. So it's very useful investigation if the coronary arteries are normal. But if they're abnormal, you don't really know whether you need to go on and do a coronary angiogram or not. So this is a case where you can see on a CT angiogram at the top left that there is apparently a narrowing in the artery. And that's narrow enough the cardiologist will want to do an angiogram. But there's an American company that has now realized that you can get much more information from those CT scans. You don't have to do anything differently. So they have developed algorithms that enable you to work out what the effect is of that narrowing on the blood flow. And the pictures from that investigation are shown on the right. So you send your, from the UK, this is now approved by NICE. You send off all the images from the CT scan, and then you get back those nice pictures on the top right. And in this instance, it would have told you to go ahead and do the angiogram at the bottom, where you can see the narrowing, and you would do an angioplasty and put in a stent. This second picture, you can see there's a a mark narrowing there apparently on the CT. When you send off the pictures and you also, via an electronic banking system, will transfer £1,200 for that information to come back. And you can see that this time it is telling you that there is a functional stenosis there, justifying you to go on and do a coronary angiogram. So this, which does not involve doing any more tests on the patients, delivers the equivalent of $4,000 in terms of decreasing the need for coronary angiography. And I think is the kind of breakthrough we're going to see more of in terms of avoiding unnecessary expensive investigations. But the area where I think technology is going to make a really massive impact on medicine is genomics. Part of the reason why I want to talk about this is some of the story does come from Cambridge. So you'll be aware that the structure of DNA was elucidated in Cambridge with input from London. I'm going to tell you about the history and where the technology is now. I'm going to tell you how it can be applied to understanding diseases, and I'm going to say something at the very end on the impact on society and medical practice. So in 1977, Fred Sanger described a method of sequencing DNA. He actually, so one of the nice things about living in Cambridge is I can cycle to work. It's literally a 5-minute cycle ride. And every day I pass the house that Fred Sanger lived in when he made that discovery and won his second Nobel Prize. So that was a way of sequencing DNA, and it involved ending the chain of DNA with a different terminator for each of the 4 letters that encode genes. It's quite laborious, as I'll come on to in a second. Only 20 years later, there were a whole series of next-generation technologies developed, and the most successful of those was invented by these 2 Cambridge scientists who spun it out as Solexa, and it's now the sequencing that is used by Illumina and is responsible for over 95% of all the sequencing done worldwide. And I cycle past Sir Shanker's house also on my 5-minute cycle ride to the hospital every day. And what they did was to come up with a way of doing sequencing where instead of doing one reaction, you do thousands in parallel, and instead of ending the chain of sequence, you put on a fluorescent terminator, take a picture with a camera, then wash off the terminator, put on another base, and then take another photograph, and so on. So it has increased, as you'll see, by many orders of magnitude, the efficiency of sequencing. And this is showing you the sequencing of different organisms. So the simplest thing was a little tiny thing that's like a virus that affects bacteria. It has 5,000 bases, and that took the best lab in the world over a year of the whole lab doing it to work out the sequence of about 5,000 bases. 7 years later, you can see, and each of these roughly took a lab a year to do. 7 years later, you could do the sequencing about 35,000 times as fast. In 1990, it had gone up another tenfold. And in 2001, for the first time, people could sequence a whole genome. And the whole human genome, which was a massive collaborative exercise, it was the first time that China had really taken part in big science. That cost about $3 billion. But the race was really on to reduce those costs. And this guy, Craig Venter, in the US, took a commercial route in parallel, and he managed to produce a sequence of the whole human genome for about 1/10 of the price of the academic project. And in doing that, he believed that you would be able to patent genes, and that's why he got the backing of $300 million. In the end, fortunately, the and this is something you might ask me questions about that I won't be able to tell you much about, but very wisely, patent regulatory systems around the world decided that although up till then it had been reasonable to patent a human gene, after that it was no longer deemed reasonable to patent something that had been produced by evolution, not by humans. And you can see that the cost of sequencing a human genome has fallen from several billion US dollars to less than $1,000 in less than 20 years. So this makes the Moore's law, which is semiconductor development, where things have improved about twofold every 2 years, and has underpinned those remarkable things like the iPhone. This makes that look like a trivial rate of development. So it is leading to an unprecedented speed of detecting and diagnosing new rare diseases. So if you went back 20 years, you could publish a new disease and the gene identify, in which mutations were occurring in any of the top journals. Now, new genetic diseases are discovered almost every week, and you'll find it hard to describe them in any journal without a great deal more work. And it's crucial to realize that many things that we regard as common diseases, with genomics, are being broken down into many rarer diseases. And that's going to have massive effects on how pharmaceuticals are marketed and how much we pay for an individual pharmaceutical. So they're going to be useful in fewer people and much more expensive. But hopefully, one of the great hopes of this is that we will, instead of giving medicines to everybody with a particular label and accepting that many of them won't work, we hopefully will target them better, and that may justify these higher prices. I'm going to tell you a quick story of my own. Most people with kidney disease don't have lots of family members with kidney disease. But interestingly, if you look carefully, although that's what nephrologists think, actually it's much more common to have family members with severe kidney disease than nephrologists realized. And we were interested in this large family in West London, which had unexplained kidney disease and a funny appearance on a kidney biopsy. And we found that this was due to a mutation in a regulator of something called complement that is part of the inflammatory cascade. And that was interesting because this gene was not previously known to do anything at all. So this not only identified a diagnosis in these patients, it also identified an important function for this gene. And we actually found that this was really quite a common problem in Cypriots. It occurred as a single mutation probably about 10,000 years ago. And an interesting question is whether actually it might have had some benefit in younger people. And it was quite significant in showing that these patients were not going to benefit from high-dose corticosteroids. So it hasn't developed a successful treatment yet. We have some ideas about treatment involving plasma exchange, and that does seem to be helpful, but we have stopped a number of patients from having toxic treatments. Now, those families were from southern Cyprus, and we had an interesting project, I think, in northern Cyprus, where we were able to collect DNA from every single patient with kidney failure. And illustrating that this runs in families, nearly 20% had affected first-degree relatives. And, of course, you'd expect some of those had polycystic kidney disease, but actually many more had family histories. And we didn't find, disappointingly, any cases of our new disease in these patients, and that may be because northern Cyprus is genetically more like Turkey and less like Greece. But we did find some mutations that are characteristic of another genetic kidney disease called Alport's. It's well recognized. But I think an important thing to recognize is that the doctors looking after these people did not think that they had Alport's. And it illustrates that once you do a whole genetic analysis, you start to uncover diagnoses that doctors have not thought of. So at the moment in the UK, we're moving from the doctor having to think of you having a genetic condition to the doctor thinking, well, maybe this is a genetic condition, so I'll do a genetic test anyway. And that throws up all sorts of interesting ethical questions. And it's the subject of this massive project. The man in there is a part of history now. He's dressed up to look like a scientist, but it's actually David Cameron who was our Prime Minister. And he chose to announce this project in the Cambridge Cancer Institute. And he said the UK government is going to sequence 100,000 whole genomes. I'm wary of saying this in the presence of a minister, but I think a science minister is okay, but the Prime Minister making a big scientific announcement like this is a bit dangerous. So there wasn't the money available to do this, but once the Prime Minister had said it, the money, of course, had to be found. Also, the scientists knew this was going to be difficult, but the Prime Minister didn't know that, so he said this would be done within a certain amount of time, and that ended up being overambitious. And this has been an important project. Interestingly, a number of countries around the world have been inspired to do this, but it has been quite cumbersome. And the idea was that the health service would do this whole genome sequencing at scale, that it wouldn't need clinical genetics experts. The information from this would be just fed back into the patient record, and general practitioners would be able to interpret the information and use it in looking after patients. Now, that's a big problem, because spread across our genomes, we are different from each other, probably in tens of thousands of ways. Now, much of those differences are in areas which we can be confident really don't matter at all, because, as you know, a lot of our DNA is junk. But actually, a significant number of those changes will be in things that could cause disease. Most of them don't cause disease, and it is really a very, very major challenge if you start out with people who are fairly normal, telling which of those things could have an effect on disease. Another problem is that setting up something like this to feed straight back into the clinical record means that it has to be done to an incredibly high standard. And that has meant that from feeding patient samples in, it is 2 or 3 years until anything comes out. And for many clinical scenarios, that is not actually useful. So because we are just up the road from Illumina, and because there's a lot of interest in genomics in Cambridge, in parallel with contributing to that national project, we've carried out some quite large-scale pilot projects to see how we can use whole genome sequencing in real time to influence patient care. And this is a project that I want to tell you about briefly, where David Rowich, who's now got a visiting professorship, I'm delighted to say, at Hong Kong University, and Lucy Raymond, have set out to do whole genome sequencing on any child on the neonatal intensive care unit or slightly older children who has an unexplained illness. So they haven't waited for the for the child to develop syndromic features. They've just said, in this population, we think there's going to be quite a high rate of genetic problems. Now, a crucial thing about this is you can do the parents as well as the child. And if there's a change in the child that isn't there in the parents, that is actually a very powerful route enriching for informative genetic mutations. They've also set this up so that they can do it on tiny volumes of blood, and so that they can have an answer back to the patient with expert clinical interpretation in 2.5 weeks or less. And so that's required a lot of work in terms of bioinformatics and workflow. This is a little out of date now, but you can see that they've done really quite a lot of children now. And this has increased the diagnosis rate by about 20% compared to existing routine practice. And in practice, in a bit less than 1 in 5 children, they are making a genetic diagnosis. Now, the reason for those numbers is that quite a lot of these children would have had a genetic diagnosis made by other routes. So in this example, there was a child who was 18 months old, and it had developed well initially, but then had a progressive loss of motor skills, and it had an abnormal brain scan. And the mother was very anxious because she was 4 weeks pregnant, and she wanted to know whether this was a genetic problem, and whether, if it was, the child which she was pregnant with might have the same problem. And you can see in green there that there's something in the child sequence that isn't there in either the mother or father. And this is a child that has inherited half of their genome from the mother and from the father. And this immediately makes a diagnosis of Alexander's disease. That's a severe leukodystrophy affecting the white matter in the brain. It's a very clear explanation of the child's developmental problem. Sadly, at the moment, there's no treatment for that. But a really important thing from this is you can see that's a new mutation in the child, so the parents can be reassured that the chance of any other children that they have being affected is minute. This is another case, and here the child was born at 32 weeks. They needed ventilation for 6 weeks, so that was a hint that something was going wrong, and they were developing severe muscle weakness and contractures. And they had lots of investigations, as children in this setting often will, but they then had a whole genome sequence, and you can see that this child has inherited something from their mother. This is an X-linked recessive condition, explaining why the child is affected. And this immediately gives you a diagnosis. It explains why the child has these muscle problems. And in this case, the baby was immediately eligible for a clinical trial, and the mother could be offered antenatal testing for a subsequent pregnancy. Now, that takes me on to pregnancy, and many of you will recognize Dennis Lowe from the Chinese University of Hong Kong. We did our PhDs at the same time in Oxford, and Dennis has gone on to have a fantastically successful scientific career. At the time when we were both in Oxford, Dennis was looking for cells from the baby in the blood of pregnant mothers. So he thought there would be enough cells there to learn about the baby. And he was able to show that if the child was a boy, then he could detect male cells in the mother's blood. And of course, a woman wouldn't have male cells with with a Y chromosome. So that was quite controversial at the time, and it did raise some worrying issues, particularly in mainland China, actually, with the single-child policy, because this was a way that you could tell that the child was a boy very early in the pregnancy. And then Dennis went on around the time when he moved to Hong Kong to show that as well as cells in the mother's blood, the placenta was actually shedding DNA. And he was able to put together a whole picture of the child's genetic makeup from these little bits of DNA in the mother's blood. That was quite a complex challenge because he needed to, of course, there's a lot of the mother's DNA in the blood, but there was enough of the child's DNA to be able to build a whole picture of the child's genome. And Dennis, along with a couple of competitors, was able to show that that picture was accurate enough to tell if the child had a major genetic abnormality like Down syndrome. And this has come into mainstream clinical practice really quickly, so it is a way that technology has affected medicine. Not everywhere. So in the UK, we haven't yet introduced this test as an approved test. You can only have it privately. At the moment in the UK, what you do is you have a blood test if you're at a fairly high risk of having Downs. That has a reasonable predictive value, but if it's positive, you have to go on and have an amniocentesis. And the trouble about an amniocentesis is that that leads to the mother losing a healthy baby in a proportion of pregnancies. And the test that Dennis developed is much better. So it has an extremely low false positive rate compared to the standard tests. And that really has saved already many children. And Dennis has also shown an interesting thing, which is that if the mother has early cancer, then you can pick up DNA from the mother's cancer in the blood before she's aware of it. And that takes me on to cancer. So cancer is a very powerful area for genomics. So cancer is profoundly different genetically from the material that you inherit from your mother and father. So it has thousands, tens of thousands of differences which have occurred by chance. And probably to get from a normal cell to a cancer cell, you need 7 to 9 key genetic events. Of course, once a cancer's developed, there's a tendency to think of it as a single evil clone. That's not really the situation at all. Actually, the cancer is an exploding population with genetic roots. So it's like humans when they moved out of Africa and evolved differently. And understanding this should allow us to detect early cancer. It should allow us to detect the key driver mutations. It will, I think, be very important in selecting treatment, monitoring response and relapse, and immunotherapy. And I thought I'd tell you briefly about a pilot project in Cambridge run by Carlos Caldas and his collaborators called the Personalised Breast Cancer Program. So this has involved every patient with breast cancer having the opportunity to enroll in a program where they will have a whole genome of the DNA they inherited and of the tumor and then multiple omics. So recruited a large number of patients. Again, because Illumina's just down the road and because we've worked hard at this, or Carlos has worked hard at this, the results are available in less than 3 weeks, and they are affecting the management in real time. They're enabling more accurate choice of drugs. They're enabling detection of women who have a high inherited risk and screening of the families. And I thought I'd share with you 2 brief stories. So the first is a 69-year-old woman, and you can see the characteristics here. Now, there was a family history of cancer, but there often is. So this woman might have been offered germline genomic testing anyway. The DNA testing showed that she had Lynch syndrome with a mutation in PMS2, and that meant that her family could be offered screening and early screening for tumors that they were at risk of. Because this is a tumor with a high mutation burden, because it's a problem with a DNA repair enzyme, there are lots of differences between the tumor and the host, so it will be suitable for immune therapy in the future. From some of her germline characteristics, we can tell how likely she is to get toxicity with some chemotherapy, and she could be assessed for multiple high-risk breast cancer genes. This is a second case, and the implications are rather similar, but the tumor is actually profoundly different. And this wasn't an inherited risk. This was an acquired risk. And we can predict that this patient would be suitable for a PARP inhibitor. And indeed, it emerged that she was in a clinical trial in which she had had a PARP inhibitor and had responded to it well. So I think this is going to profoundly affect the way cancer is managed. And this is summaries of the outcomes so far. So in 10% nearly, patients could be advised on toxicity. In 20%, they were referred to the genetics clinic for advice to the family. In 6%, it changed the type of surgery, going for more radical surgery in patients with a high risk of recurrence. Change future treatment in many patients, and current treatment in about 15%. Well, one of the areas where genomics is most important is in your response to drugs. So we haven't evolved in the context of these chemicals that doctors are exposing us to, and there's enormous variability in how we respond to those. And this really does have implications for some medicines that we're using today in terms of what the right doses to use are and what the likelihood of toxicity is. So many of you may be on statins, or when you're as old as I am, may be offered statins. A serious problem with this is some patients get muscle pain and inflammation, and there is quite a common genetic variant. It's a single change in a single letter of the DNA. If you have both copies of that variant, then that increases your likelihood of getting muscle pain with statins by about 80-fold. And the test can be done for a few pennies. Infectious diseases are rather different. So here we're thinking about the genome mainly of the infectious agent. And this enables us to track infectious agents with absolute precision and to understand in a patient how they're evolving and whether, like cancer, there's a genomic diversity in the patient. And this is a brief story from Professor Floto in Cambridge, who's a respiratory physician, and he's interested in a close relative of Mycobacterium tuberculosis, which causes TB. And the organism he's interested in is called Mycobacterium abscessus. It's not a problem for all of us in this room, but it's a major problem for patients with cystic fibrosis. And you can see that it shortens effective lung function, so the time to death or a transplant, by about 25 years if you have Mycobacterium abscessus. So instead of needing a lung transplant at an average age of 40, you'll need a lung transplant at an age of about 15. So it's a very serious infection. It's increasing worldwide, it's multi-drug resistant, and it's very poorly responsive to therapy. And, sorry. It's obviously a rather big image. I must just be patient, I think. So the view has been that individual patients with cystic fibrosis get colonized from soil or from water where this bug is common. And by sequencing the individual organisms, you can tell how closely related they are. If individual patients around the world have picked this up from soil and water, then the organisms will be highly divergent. If they've picked it up from common sources, then you'll see clusters that are closely related. And if it's spread between patients, then you'll see that they're very closely related. And this enables us to see how this might be spreading in a hospital, which would obviously be an important problem if people are picking it up from each other in the hospital, not from the soil or from water. And this is showing you a single room in a cystic fibrosis unit, and once the patient has a red dot, they have Mycobacterium abscessus. And these 4 patients, over a year and a half, each went into a room in the hospital. And the first patient who had Mycobacterium abscessus went into the room, and then you can see the other 3 patients, when they were looked after in that room, each developed Mycobacterium abscessus. And you can see that isn't because the room wasn't being cleaned, so each time there's a blue line there, the room has been cleaned. And the genomic analysis showed that this was a single clone that had spread from that first patient to the other patients. And this is because the organism is actually highly resistant. They've done nice experiments showing that when you cough, this travels about 20 meters, that it deposits in the room, and normal cleaning won't remove it. Actually, the hospital where our respiratory patients are looked after is called Papworth Hospital. Interestingly, it was developed as a TB hospital for normal people about 100 years ago. But of course, now TB is normal TB in normal people is effectively treated. And that was what the hospital looked like in 1918. And that's what the hospital looks like today. You can see it's hardly changed. And it is not an easy hospital in which to control airflow, clean rooms, ideally. But fortunately, we're building a new hospital on the main Cambridge Biomedical campus. I hope you'll come and visit and you can see this new hospital. And because this research was done while the hospital was still being planned, the layout and gas exchange of the cystic fibrosis unit was changed. So as a result of these genomic analyses, we changed the layout of the unit to decrease the chance that patients would catch the infection from each other. Now, a really striking thing is that if you look at different units around the world, actually the Mycobacterium abscessus is related to each other in all those units across the world. And it seems to be a small number of clones that are spreading around the world. And what Andreas believes is happening is that people like us, scientists, clinicians, nurses who are researchers, get colonized by the patients. We're asymptomatic because we have a normal immune system and normal respiratory system. But we then go to conferences and talks like this, and it appears that we can colonize each other and then take this nasty bug back. Now, this has not been a popular finding in the community because it does have implications for respiratory physicians looking after these patients. Now, understanding the immune response is another fascinating area of genomics. So it's important to realize that although we're born with one set of DNA instructions, our immune response is actually developed in real time from the moment we're born. And we can make billions of different immune recognition molecules. And that's why once you're vaccinated, you're able to develop an immune recognition of a nasty organism and not get a serious infection when it comes back to you. But our immune system's very potent and it can go seriously wrong. And one of the examples for me as a nephrologist is called vasculitis, where you get inflammation of small blood vessels. And shown here are graphs of kidney function in two patients who were indistinguishable at the time when they presented. So they both needed dialysis. The one on the left with the red treatment symbols had every treatment we could think of, and they still had a relapse of the disease because their immune system, we couldn't switch it off and stop it from inflaming their blood vessels. The patient on the right, who was indistinguishable at the time when they came to see us, had a single course of disease-controlling treatment, and their immune system was then effectively reprogrammed, and they never had another relapse. So how could we tell these patients apart? And Ken Smith, who's our professor of medicine, has led a brilliant project that I thought was stupid when I read a grant proposing this 10 years ago. I'm very glad that he ignored that. He got the grant from somewhere else. I haven't told him, by the way, that I was involved in not giving him the grant. So please don't tell him that. But I think actually it helped him think back more clearly about it. And what he did was to look at the gene expression. So these genomic technologies enable us not only to look at the genes, but also to look at where genes are turned on with complete accuracy. And Ken asked whether patients who respond well to treatment and patients who respond poorly to treatment look different at the time when they come. And I hope you can see that so each of these columns with red or green in is an individual patient. And so in the left-hand thing, I hope you can see that there are two rather different patterns of turning on and off individual genes. And so this developed an idea for a signature that would tell you whether patients were likely to respond to treatment or not. And then he did another cohort, and you can see that this was able to predict which patients would respond to treatment or not. And this is really quite powerful in a range of autoimmune conditions where you either can turn off inflammation easily or not. So this is in vasculitis, and you can see that if you have one kind of signature, then your chance of going 3 years without another flare is about 10%. If you have the other signature, it's about 80%. And Ken is now doing prospective trials to see if we can alter treatment on the basis of that signature. I'm towards the end of my talk now, but this is also a very, very powerful way of looking at other species. So in understanding why some types of crop, like rice, are better at yielding food or more resistant to disease. And it's also useful for understanding animal health. And Steve O'Reilly runs our Institute of Metabolic Science. He's very interested in obesity. There's a problem with studying obesity in mice and rats because they're rather different from us in their metabolism. But Steve was interested in dogs, which are a bit closer to us. And he was interested in Labradors. So Labradors, I don't know if they're popular in Hong Kong. I haven't seen many dogs here. They're a very popular breed in the UK because they're affectionate, intelligent dogs. But there is quite a big problem. They are massively inclined to get overweight, and they're very good at looking hungry. So most of their owners give them food, and they get seriously overweight. And Steve was able to show that there's a single mutation in Labradors that really has a major effect on body weight. Now, like humans, like many genetic causes of obesity in humans, this is the desire to eat and the inability to feel satiety. So Labradors with changes in this gene are really hungry and really good at persuading their owners to let them have more food. And it's interesting to wonder, so these are called Labradors because they come from the east coast of Canada. And it's interesting to wonder whether this was actually selected for by dog breeders. And the answer is probably yes, perhaps for two different reasons. First of all, the dogs were involved in fishing, and dogs that are fat float better and can swim more easily. The second thing is this genetic trait is actually more common in dogs that are used as guide dogs for the blind, and it probably makes them easier to train because we basically train dogs by giving them food rewards, and dogs with this trait are probably much more interested in the food reward. So you can see that when breeders were selecting dogs for trainability, this might have been a really helpful trait. But as a model of a particular kind of human obesity, this is a model that you can't do in a mouse because a mouse does not have this gene, POMC. I'm nearly at the end now, but there is a massive revolution which I'm not really going to touch on, which is we can now edit the genome with complete precision due to a series of discoveries which exploit a natural system for precisely cutting DNA. And that means we're entering a new era where we can actually change DNA more or less at will. Now that's going to be very important for treating human diseases. There are lots of ethical issues there which we might want to discuss. But also it offers huge potential for improving crop yields and disease resistance. And there's some very elegant approaches using a gene drive which transfers this gene into normal DNA as well. you can do things like eliminate a particular species of mosquito by engineering a system where the females are completely infertile, and you rapidly end up in a situation where there are only male mosquitoes, and that is the end of the mosquito world. And you only need to introduce about 10% of the wild-type population to do that. So not trivial, but major implications. And that takes me on to doing things that evolution can't do. So in the Laboratory of Molecular Biology in Cambridge, there are people working on a genetic code that, instead of using 3 letters and encoding 20 amino acids, uses 4 letters and can use a couple of hundred of amino acids potentially, opening the way to doing things that evolution could never do. But I want to come back to medicine, and this is something Ron and I have discussed in the past. So there's been a sort of dream that if we can share genetic information with patients, that will help us enormously in changing their behavior. And Teresa Marteau, who's a social scientist in our School of Clinical Medicine, did an interesting experiment where she asked, if I give people genetic information about their cardiovascular risk, does that help them to make better decisions? This was a randomized trial. Unfortunately, the answer is no. It's no better than giving them non-genetic information, like you should take more exercise or you really should stop smoking. And I can see Ron looking highly skeptical because Ron is a rational being. But I think we all know as doctors that when we're patients rather than doctors, changing our behavior is not easy. And I think as a wise doctor, the most difficult thing is probably helping patients to take wise decisions about their future. And we haven't yet reached an era where genomics, I think, is really going to help us do that. This is a picture of, this isn't my research laboratory in Cambridge. This is Trinity College, also founded by Henry VIII, where I'm a fellow. And I think Ron was actually a graduate student. Is that right, Ron? An undergraduate student. And I'm delighted to answer any questions. Thank you very much.
Thank you so much, Professor Maxwell. That was a really riveting talk. I now know why I am, float so well. And I'm now more conscious of the legal liabilities for organizing colloquiums for the exchange of Mycobacter abscessus. Perhaps we do have about 10 minutes for questions for Professor Maxwell. I ask you to use one of the microphones that are being passed around and to introduce yourself, your name and your affiliation. Yes, Professor Fukuda.
So, Professor Maxwell, thanks. That was a fantastic talk. You know, right now, the technology is exploding in a number of different realms. And so in this talk, you're really focusing on the genetic technology, which has really revolutionized a lot of our thinking about things. But you know, the technology is advancing in terms of imaging, as you pointed out, with radiology. And so the implications for that in terms of diagnosis of a number of different areas, not just in radiology, are profound, as are the algorithms for making decisions. And when you take a look at the span of where we're seeing technology change, where do you expect to see the greatest impacts over say, the next decade or so? Where do you really see us
Well, it's a question I could answer in several ways. So one question would be, which bit of the world do I think this is going to happen in? And I don't think it's going to be led actually in either Europe or the USA, because I think our health systems are actually quite inert and conservative. And I think mainland China is very interesting from this perspective, because people, the rate of using digital devices to do new things is really rapid, and the healthcare system is really changing very fast, and at least as I understand it, is very consumer orientated. So watching the rate of new hospitals being built in China, and the rate of change in other ways, I think some of these areas are going to be explored much more quickly in Asia than in either the US or Europe, and particularly in mainland China. What ways do I think technology is really going to change medicine rapidly? Well, the hope is that we will make diagnoses and risk stratification more cheaply and more wisely and better. I would have to say that I think this is a difficult area. I mean, particularly in places where people pay for things and medical systems make money out of patients. It's easy to persuade people to pay for things that they might actually be better off without. And I think this is going to lead to an explosion of new things that you can pay for. And it's quite likely, I think one of the successes of modern medicine has been restraining that. And although we might have many concerns about the way healthcare is regulated, a very good thing is that it has in general meant that consumerism is not unfettered. And I think, this may, in one version of the future, I think healthcare will become much more consumer orientated. And I think that's an area, where particularly public health experts would have a real concern. I suppose one of the areas that I would see a particular opportunity in is in new non-invasive tests. I mean, particularly blood tests, where most of the blood tests we use were developed probably, 30, 50 years ago. And we haven't yet learned a great deal more about how to use blood tests to decide on treatment decisions. And I think that's an area where genomics is going to have a massive impact very quickly.
But Terry, can I ask a follow-up question? Sorry, I don't want to hog it, but given that we are such a consumer-oriented world, and we are in love with technology, and innovation is used in every other sentence and so on, do you think that the ethical frameworks that we have in place now and our ways of interrogating, is this a bad thing to do or a dumb thing to do, are robust enough to protect us from our own arrogance?
No, I don't think so. And I think we need much more dialogue between, people with different perspectives. I think as doctors, we make many assumptions about patients that are, based on our own prejudices. I think the way we regulate medicine is, a complex evolution, not quite as complex as the way a cancer develops, but I mean, it's very complex, and I think it is not well placed for the next, decade or two, which makes it a very exciting time to think about these challenges. And it's one of the reasons why I was so pleased when Ron asked me to give this talk, because I think it's really important we think about these issues carefully. One of the things that really strikes me is the difference in the way we think about informed consent for Google looking after our data versus informed consent for a hospital looking after our data, or taking part in any kind of medical research. So when Google give you a form to sign on your laptop, I mean, some of you are lawyers and you probably read the thing that you are consenting to. I believe that it is not designed to make it easy to read. I think the whole thing is designed to make it as easy as possible to click on the I accept box. And in medicine, we certainly can't do that. I think that's a better position, but we do make it a bit too difficult, I think, to participate in some kinds of medical research that I think as a society people would broadly value. So I think it is too difficult to wisely collect the information from a whole health system and then take good decisions as a society which affect individuals.
That was Professor Fukuda, who is the director of the School of Public Health at the University of Hong Kong. Yeah.
Professor Maxwell, while I'm a pediatrician, I just want to go back to the topics of rare disease. And I'll just share with you some experience I've had while practicing as a doctor in mainland China once a week for the last, 6-7 years. And indeed, China is very sort of much driven by consumers in terms of ordering the genomic tests. Because rare diseases are rare and hence might not be recognized by pediatricians and doctors. So parents of children with rare disease always have this, odyssey of getting the diagnosis. So a lot of them now, in fact, as low down to the county level, they will be able, because, big sequencing companies, they're running commercial, will be able to tell, quote unquote, through whatever agents to parents that you could send the blood sample and then they will get a result back. And indeed, rare disease like ADA deficiency, extremely rare, immunodeficiency has been diagnosed that way. And then it really short-circuited the whole training process for medical profession to understand rare disease. And rare disease got about 5,000 of them, and I gather in Europe, the estimation is 6% of Europeans actually had rare disease. Yeah. So in fact, it's not rare when you put them all together.
Absolutely.
But in order to train doctors to understand, recognize, order the test for that rare disease is next to impossible. So I think the parents really taking the destiny into their own hands. So for God's sake, I've already lost a child with this God knows what mysterious disease. I'm going to take it into my hand. And therefore, they spend that 3,000 or 5,000 renminbi and get that test done. And then ended up in another hospital. Oh my God, what is this? And then they start reading of course, the standard of the report is far from ideal as compared to the one sort of, in Illumina or from Boston. But I think the mainland Chinese parents are really taking the destiny into their own hands because they're fed up. They're really fed up with this diagnostic odyssey. So that is one sort of comment I want to share with you. So my question for you is, since 6% of the population had rare disease, and it's so difficult for us as medical profession to really do that diagnosis, what do you think is the strategy to get to the bottom of this 6 or perhaps even 8%? I think the high bound is 8%. It's not a small number,
Well, I think things are going to change very fast, and my guess is that within about a decade, we will have overcome this barrier, and basically everyone will have their whole genome sequenced. And then the question will be how to use that information. So, and at the moment, I think the kind of question that is difficult is if I found that you had an ApoE4 allele that gave you a higher risk of dementia when you were older, would you want to know that or not? And once I know it as your doctor, how do I decide whether to tell you or not? Or if you send it off straight away as a consumer test, how should the company share that information with you? And if it's, say, Huntington's, where we have no current treatment and the situation is horrible, is that a good thing to know or not? So I think we have got quite a bit of thinking to do on that. But my own view is that medical knowledge can never be held back. As you say, this is short-circuiting the way that the profession works at the moment. And I think we should embrace changing much more rapidly. One of the most striking things is at the moment in many countries, including the UK, to have a genetics test, you need to see an expert geneticist, and they'll give you careful genetic counseling, and the whole system regulates genetic testing. It's pretty clear to me that in every specialty you'll have to have people who are able to tell whether the genetic information is going to alter the management of that patient. So in every team of specialists like nephrologists, my specialty, I think there'll have to be at least one, probably more, maybe everyone will have to have serious genetic expertise.
Well, I think we should close the lecture here at this point. I think we have already run way over time. It leaves me to ask you to thank with me Professor Patrick Maxwell for speaking to us this evening. On behalf of the Center for Medical Ethics and Law, we also wish to thank our partners, the WYNG Foundation and the Hatton Trust, who have been supporting us for all these years, not only in this series of lectures, but in the work of the center. Well, just a bit of advertising. I think most of you would have already received one of these. So if you're minded to study some more, especially the law, please get yourself one of these brochures, if you don't have one, from the table outside. And you should know that over the next 2 days, we'll be running a conference on property in the body. So if you want to know what is the law in Hong Kong in relation to unclaimed fetuses, unclaimed dead bodies, IP claims to the living and the dead, as well as return of benefits to research populations, please do also sign up. We accept last-minute registrations, and it's free. And all you have to do is really to scan the QR code here, and you have all the details of the conference. Thank you, Professor Maxwell.
Thank you. Thank you very much.